Hi everyone, welcome to the latest SPI how-to video. On today's video we're going to be covering how to fill in the DNA application forms and also registration forms. You do need to do both of these at the same time, and so let's get started. What you will need to DNA and register your calves. Obviously you'll need a sample of some sort to get DNA from. TSUs are preferred. Hair samples on approved Neogen hair kits or semen samples. Just be aware, TSUs are now the preferred option for Neogen, and hair and semen samples are attracting an extra fee. One thing to remember when you're purchasing your tissue samples and the gun, I'll just show you here. They come in two forms. So here they're in a box of 10, individually numbered, obviously. And that number, when it's scanned at Neogen, stays as that number. The alternative is purchasing them in a kit with obviously an ear tag and an NLIS number. Now the difference with these is when this little barcode on the end here is scanned at Neogen, it actually comes up with the electronic ID number that relates to your NLIS number. So it can cause some confusion, and it is up to you to provide the electronic ID number as well as the tissue sample number if you are using this form of tissue samples. So they are now the preferred method of extracting DNA. What else do we need? Obviously, if they're embryo calves, we need our implant data. So the implant sheets from the vet or your own implant data. You need joining and carving records, your paddock book, spreadsheets that you've got with joinings on them, your cattle program, whatever it is that you use to keep your records straight. And the more information you have at hand, the easier it will be, and the more likely you are to get a positive parent verification from your DNA application. Obviously, we also need the DNA application from the SPI website and the calf registration form from the SPI website, both of which can be found here. Now, I went on and downloaded them a little earlier. So this is your Neogen Australasia cattle genotyping application form that is specific to SPI. And you will also need the SPI calf registration form. So let's start with the DNA application form. So here we have Hairy Speckle Park, 123 Hare Road, Harryville. The breed is Speckle Park. Our herd prefix is HAR. My apologies if that's yours. We're just making up an example for this. This is a form that needs to be completed on your computer. If you are wanting to still do manual DNA applications, I recommend that you ring the SPI office and find out the best way to do that. Okay, so coming down to the form down here. So we've collected tissue samples from all of our calves born this year. This video was made in 2019, so they are Q calves, and we have our tissue samples. The tissue sample number, which again, just let me be clear, there's a number on the actual physical tissue sample which you will put in this box. So that's usually 2 letters and then 7 numbers. Again, this is just for the purposes of showing you how to fill in this form. If you have purchased tissue samples as a package with your NLIS tags for the year, then you will need to provide Neogen with the IED numbers, electronic identification numbers from those. Allflex should give you a file, what they're calling a dump file, in order to give those then to Neogen so they can correlate those. So our first calf that we are DNAing and registering is HARQ1. She is a female and she was born on the 12th of the 2nd, 2019. Now, as most of you will be aware, in order to get a calf that was born after the 1st of January 2018 registered with SPI, they require the SPI bundle, which is under the SNP system of DNA. Over here, you will see that the bundle is already listed. That includes the GGP50K, PV, that's for parent verification, POL, myostatin, coat, and leptin has just been added this year as well. Parent verification in this bundle is only possible if both the dam and the sire are on the SNP system. Luckily for a Q1, they are. So all we need to do is the drop-down menu, click on the bundle, tab over. Now, I know from my implant records that this calf is Putnam GNK5X. They are my implant. That's from my implant records. That's the recip that the embryo was put into, and that carved out with that embryo. I am, pretty confident in my paperwork in this. Would I, as a matter of safeguarding, provide optional dams and sires? Hell yeah, I would. Because something could have gone wrong in here. Some paperwork, they could have been mothered up wrong. Whoever was doing the paperwork on the day has put down the wrong ident for the wrong recip when the calf was born. There's a lot of things that can go wrong. So yes, I would, if I was doing this, I would say, yes, there are optional dams or sires, and down here you will see there is the opportunity to put an optional list of sires and an optional list of dams. Now, the calves that we're looking at today were all implanted at the same time. There were 30 embryos implanted of 5 different combinations. So I would, as a matter of making sure I got the best chance of a parent verification, put all of my combinations of embryos that I had implanted on the optional list of sires and the optional list of dams. Now I'll come back to that because we want to put the sire case number or barcode in here and the dam case number or barcode in here. So where do we find those? You head to the SPI website and click on Inquiry, and this will take you to the Speckle Park Inquiry page. The easiest way I found, particularly if you know the animal's identifier, is to go straight to the animal. So we know that we are using GNK61Y as our sire, Kodiak Putnam. Click on that, and here are all of his details, including DNA typed and SNP typed. Now the difference here is that the DNA typed is for the MIP system, and the SNP typed is for the SNP system. So we're right to get this calf parent verified in the SNP system. So we're going to take this case number, easiest just to copy and paste, go back to our form and paste it in there. You can also type it in, obviously. I actually find when I'm doing this at home, having two screens for your computer is really, really helpful, because you can go back and forth and have lots of pages open without having to sort of jump from one to the other. But for the sake of this webinar, we've got one screen. Okay, so we're now going to look for GNK5X. So again, we're going back to the ABRI SPI database, typing in GNK5X. Just make sure that you're putting the space in between here, because otherwise it won't recognize it as 5X. So again, 5X has a MIP profile number and a SNP profile number, and we're taking the SNP one today. So we're copying, coming back to our DNA form and pasting it in there. Now, just coming back to this optional dams or size, look, you could be 100% sure that the animal that you're registering and parent verifying to is the progeny of these two animals, and that's fine. I have found with the hundreds and hundreds of animals that I have DNA'd, and particularly in the last 2 years where parent verification makes or breaks the registration, and because it's now all going through the society, that the more options you give in your first application, one, the less stress there is when you get a result that you're not expecting, because you do get some sort of result. And two, it actually costs you less, because whatever optional list of size or dams that you provide will be tested as part of all of this initial testing. If you don't give any optional dams or size and you get back a negative parent verification, and you have to then provide information about the dam and the sire, you will get charged a re-parent verification fee, which I think is about $11 plus GST. So it's worthwhile taking the time to go through your implant sheets and your joining data and all that, and providing in your original application the optional list of size and the optional list of dams. The reason that you must remember to put yes here, if you are providing a list of size or a list of dams, is this lets the SPI office know to look at these two forms and to provide those to the lab so that you make sure that they get tested. Okay, so let's fill these in. So just again, pulling out some idents. So in this implant, we had embryos from GNK46Y. We had embryos from MBG25U, and we had embryos from LMT01B, and embryos from GGG68L. So again, just pulling these out. Now we do need to provide the case number for these, because this goes directly to the lab and they grab these case numbers. They don't necessarily grab the ident. Had a problem recently where I had made a small typo in one of the case numbers, and they had not necessarily compared it to the tag number that I had provided, and so just tested against that case number I had provided, which ended up being some random Brahmin animal. And so that came then back up on my results, and obviously didn't affect the result either way, apart from I got a very strange excluded number. So very important to double check your case numbers. That's why I find copying and pasting is the easiest way to do it. Now, you may have spent the time collecting your own case numbers and have your own spreadsheet to be able to do that. Fantastic. For those that haven't, the only way that you can get them is going back to the SPI website. So again, we're just going back and forth. The beauty of using the SPI database is also that you are double checking all the time that the animals that you are wanting to test against have the necessary permits to be able to have their calves registered. So here up to Specs Ulysses 25U. Again, our SNP type, copying, jumping back onto our form and pasting in there. So we'll just quickly do the other two size, and then the process is basically the same for our dams, for our implant. And again, you may have a much better system than this. This is just the one that I use to ensure that all of the information I'm providing to the society is as accurate as I can possibly make it. And last one. Beautiful. Okay, so for our embryo calves that we've implanted, obviously we've only got one on the form at this stage, Q1. We've got another 30 to put on there, and they will have their own dam ID and sire ID. But just to give you the best coverage, I guess, on getting a parent verification result that you're expecting, always putting in your optional list of dams and sires. So we'll just quickly type in some dams here. These are ones I'm familiar with. Again, you will have your own combinations and joinings. This is purely for an example of how to go about it. Okay, so we've got 4 sires but only 3 dams. Again, we would just go back and forth to SPI and type in their ID and get their SNP bundle number. Okay, so great. Now we have all of our optional dams and their case numbers, and our optional sires and their case numbers. Now you will remember these 2 sires and dams were part of our implant as well, and if we were coming down here and we were wanting to DNA HARQ2, who was from the same implant but a different joining, was born on the 13th of the 2nd, 2019, a bundle. We know that all our dams and sires have SNP profiles, sorry, and we are wanting to give ourselves the best option to have a positive parent verification result. So Q2 happened to be a combination of, let's pick this guy, GGG68L, and he was with WGS L221. So we'll just pick up her case number. So this, again, look, we're 90% sure, we're 100% sure that this is the combination that this calf will come back. But in the implant, we had this combination up here, 5X61Y as well. But they're not listed on our optional sires and dams at this stage, and they need to be. So I would, again, copy from here, type in here G&K61Y, get his case number. I would do the same with G&K5X. And grab her case number and paste down there. Okay, so we've got our two ET calves on our system. We've got all the information that the lab requires to parent verify them to our expected dam and sire, but we've also provided an optional list of sires and an optional list of dams that were in the same implant, so that if something has gone amiss in the paperwork from way back last year when these embryos were implanted, all the way through calving, all the way now to actually having a tissue sample ready to send, something could have gone amiss, but we've given ourselves the best chance of finding out who the animal is and having parent verification in place so that they can be registered. Okay, so what if it is a natural calf? Well, the system is the same. So we have a tissue sample, we have the next tissue number, we have it was the third one born, and it's a girl, and it was born on the 15th of the 2nd, 2019. So now we require the bundle to get this calf registered. Optional dams and sires. Now with natural calves, look, there's probably less chance of there being a paperwork stuff up, but it does happen. And there's also situations where, you know, a bull breaks down and so you throw another bull in, and the timing could be, you know, a day here or a day there as to who the actual sire is. So look, I would still put optional dams or sires in, or sires at least. I mean, with a natural calf, you know, with people getting tissue samples and tagging calves, there is far less likely to be an issue on the dam side with natural calves. And you will know from your own paperwork whether the sire thing, whether there's a couple of possibilities, or it is, you know, you've used the one sire for joining, or, you know, if it's an AI one, well, you may want to put the AI sire in here and put the backup sire in the optional list of sires, particularly if the dates are a little bit close. So that part is up to your own program and your own confidence in your paperwork. But I can't stress it enough, the more options you give as far as dams and sires, the more likely you are to get a parent verification out. So our natural calf here is actually an AI calf, and we AI'd this cow to this guy, and the cow was So she's had a natural calf again, and I know I'm repeating myself, but repetition means that things get stuck. So JKH 300X, spots and sprouts, stands alone. Again, he's MIP typed and SNP type, and he is AI approved. So he has everything in place that we need to register this calf if it is a AI calf. And then our natural dam is WGSL225. So again, SNP typed. She is an ET donor, but in this case that doesn't actually matter because this is her natural calf. Well, let's tidy that up. Beautiful. Okay, so she was AI'd. The backup bull went in 2 weeks later. The dates are, close enough that I would definitely put our natural backup sire down as a possibility. So he was, in this case, M34. So we're then again going to look him up. This is not an advertisement for our animals, I promise. This is just purely for showing you how to get stuff done. So you will notice that this guy is not an ET donor, an AI approved sire. So in this case that doesn't matter because he is only the backup natural sire. But if you come across that and you're trying to register an AI calf to a particular sire or an embryo calf and they don't have the AI approved or the ET donor in the cow's case, then you need to do some research and either go back to who you bought the embryos or semen off, or organise those permits yourself. Okay, so then we're back to this. So there's just one more thing here that I want to show you. So this is our, this is a calf that is needing to be registered, obviously. HARQ4 is male. And it was born on the 16th of the 2nd, 2019. Again, all examples only. So this calf has to have the bundle. But this was actually just, according to our records, this was a calf that has come from an embryo that was basically left in the bottom of the tank. We had one recip left over, and this embryo could have been 5 years old, but hell, let's throw it in. We've got a recip there. And according to our records, the embryo is from GGG54R and the sire is BED50T. So again, we then need to find the case numbers for GGG54R. So we head back to the SPI website, type in GGG54R, and here she is. So she's an ET donor. She only has a MIP profile. So we need that profile number. But when we come back to here, we will see that because we have only requested the bundle and she only has a MIP profile, we will not get a full parent verification with just the bundle. So again, coming to your drop-down box. So this animal, and there are a few older ones that are still just on the MIP system, so it may look like doubling up, but the only way that you're going to get your calf registered, if they have a dam or a sire or both in the MIP system, is to request the MIP plus PV. So what about 50T? What has he got as far as MIP or SNP? So we go and check it out as well. He is AI approved. Yes, he has a MIP profile and a SNP profile. So you could do this either way. You could just get your complete MIP parent verification done with your MIP plus PV using this case number. Or if you wanted to, you could get the sire verified in the SNP and the dam verified in the MIP. Now, there are some cases where that is necessary, when the sire only has a SNP profile and the dam only has a MIP profile. I think I prefer, if there is a MIP possible, to get both done in the MIP system, and then you know that that combination is 100% correct. We have here, because this is part of the same implant as our first two calves here, we've got all of our optional lists of sires and our optional lists of dams. They're ready to go. So then you would just continue on. If it's hair, obviously you put hair in here. You don't need a TSU number if it is hair. When you have completed this form with all of your animals, the form needs to be emailed to SPI. The email address is here. And the samples need to be sent with a hard copy of this form. So print off the form and send that along with your samples to the SPI office. Now, the samples need to be in a container or the box that they came in, or you can get trays, blue trays with the tissue samples go into. Please don't send a Ziploc bag of tissue samples. And please, please, please make sure that everything on here, these tissue sample numbers, the animal IDs, correlate with the actual samples. If you're doing hair, then hair samples need to be on approved Neogen hair sample cards. Ziploc bags with hair samples that have no associated paperwork or are not on Neogen sample cards will not be accepted by SPI. They will be sent back to you. We are working really closely with the lab to reduce times down for all of this testing as much as possible and to make the system really streamlined, and the society needs your help to do that. So do the work at home, make sure you keep good records and put all of the information that you have on these forms and print them off and send them with your samples, and make sure that your samples are in the appropriate format so that the hair samples are on Neogen approved hair cards, your tissue samples are in, in boxes or in the blue trays that you can purchase. Take the time to do that stuff at home, and that saves time then for the secretary and ABRI when they're then collating those to send on to the lab. That's it for the form for the DNA. So any questions on that, please email SPI. So the next part of this is registering the animals. You have to fill in the SPI calf registration form at the same time and send it in at the same time as your DNA application form. If you just send DNA forms without the registration form, then it short circuits the system and takes a whole lot longer because the ABRI staff actually have to do this part for you. It's not their job. Your job as a stud owner is to look after your own paperwork. So we have Q1, Q2, Q3 and Q4. Our herd ident. is HAR, and we've got 4 calves that we are registering on today's form. The calf herd is HAR, calf tag is Q1, Q2, Q3, Q4. The dam, our sire of our calf, is these guys, as far as we know. So we'll put them down here. What happens if these optional list of sires or dams actually turn out to be the correct dams and sires of our animals? What happens to these details? Well, the system automatically updates them. The one thing that you will have to be aware of is if you name your animals based on your expected dam and sire, and those expected dams and sires don't end up being the parent verified animals, then you may need to change the name. These are embryo calves, all of them. This is an AI calf. The mating code is A for AI. Now, date of birth, it is 2019-02-12. So it's 201-02-13. So 2019-02-15. 2019-02-16. Now, female, male, female, male. None of these guys have been twins. This is information that is used for breed plan data. So all of these calves were easy, unassisted. If you're needing to know what each of these things mean, the instructions are over the page here. So calving ease score, not recorded, 1, no difficulty, 2, easy pull, 3, hard pull, etc., etc. If you have a lot of animals and you have ET calves and natural calves, or however you group them, this is where you group them for breed plan data. We're now into birth weight, so all of these animals were recorded at birth. We had 36 kilos, 38 kilos, 40 kilos, and 35 kilos. K for kilo. Here is our colour. So we have white here, speckle, definitely cool, leopard. Now, even though we're testing for the pole gene, you can still fill this in if you know is polled or scurred. Obviously, if it's horned, you can't register it. So we have 3 polled and 1 that has scurs. Okay, now you remember that 3 of these were embryo calves, these 3. So our recipient ID is, look, you will have that on your paperwork. If it's 521R and the year that she was born, and whether it's an Angus or whether it's a Charolais, or however that works for you. In our case, they were all Angus. Okay, so then we're naming our animals. So you don't need to put in here hairy, whatever your name is, or your stud name beforehand. That automatically happens in the system. So you just pick the name. Now, for some of you, that will be the tag number of their dam and sire. For some of you, it will be using the letter of the year. So in this case, Q for Quentin, or you will just have a wonderful random selection of names. For today's case, I'm going Clary Q1. We have Muchness Q2. We have Missy Q3, and we have Macho Q4. Their breed is Speckle Park, which is SK, and the year that they were born was 2019. So you can just copy all the cells down. Okay, so that is our registration form filled in for our 4 calves. We've got our expected dam, our expected sire. They are either embryo or AI calves. If you had a natural calf in here, then the code would be P for paddock, and the details would be the same. A natural calf obviously won't have recipient details. My apologies, this should be just yes, not ET. Twins, obviously you would put the details in here, and I think that's it. So you would then save this file to your computer along with your DNA application. Save them, give them a meaningful name so that you know where to find them when you need them, and then you would email both of those forms together to the SPI office. Both of these forms get sent together. You send your samples with a copy of this form, a hard copy of this form, to the SPI office. The details, let me just open this up again. The details of where to send it are here. ABRI, University of New England, Armidale, New South Wales 2351. We put those in the post. My recommendation, again after a lot of experience and some scary moments, is to either purchase tracking or express post it so that you have tracking for your samples. Okay, so you've sent your samples to SPI. They will then come back to you. You will get an email saying with a DNA sample acknowledgement, and this will have whatever testing that you have requested for the particular animals and the society reference number. Handy to keep that society reference number if you ever have to go back to them and ask questions about the results. Then on average now about a 6 weeks turnaround, you then get your animal genetic test results report. This may come through in bits and pieces depending on the system, or they will hold off until all test results are in the system and send you a report. So here you can see these are a couple of old animals that we had tested. The sire has qualified, the dam has qualified, the poll results are here. The myostatin results that you are looking at are the NT821. They are the only myostatin genes that relate to Speckle Park, and you will either have free or you will have carrier. You've also got coat here, which there's a combination of codes that you will have to say that this is homozygous black. There is a key to the results on the SPI website. You can see here that no results were obtained for M127 for coat. That could be a matter of there not being enough hair in the sample. These would have been done on hair, or the sample was somehow damaged before it got to them. For these older animals, not such a big deal. For newer animals, if you're missing one of those bundle results, whether it's coat, poll, or myostatin, you do have to do a retest, and there is a charge involved. So if you are still using hair samples and planning on using hair samples, I recommend you have a really good, clean sample and lots of hair follicles that get to use. That's it from me. Just a reminder then what you actually will need to get started, and where all the information is from SPI as well. The ladies at ABRI are really helpful and keen to help you get the best result from your DNA and registration. they just request that you take the time to make sure that you've given them as much information as you can on the appropriate forms, and that the samples are packaged appropriately so that the system can run really smoothly. Any more information that you need, head to the specklepark.org, which is the SPI website, or give the team a call. That's it from me. Thank you very much.